Detection of plant virus means to determine whether a particular virus is present in the plant sample or not. Whereas diagnosis means to diagnose or to identify or confirm a specific virus, whether that specific virus is present or not. So we will identify that particular virus. By the detection technique, we already know that a virus is associated with a particular plant sample. But by the help of diagnosis, we will identify and characterize that particular virus. So in this video, we are going to discuss about detection and diagnosis methods used for plant virus studies. So major objective of detection and diagnosis of plant viruses are: first, the early detection of disease. So if we can detect that some viruses are associated, we can predict that the disease is going to occur, and we can do some preventive measures. Next, by the help of diagnosis method, we can identify a particular virus. Then third, it is helpful for certification of planting material. So by detection and diagnosis method, we can check that whether some viruses are present in a planting material or not. If it is not present, then we can certify that the particular planting material is free of viruses. Next, it is also helpful for epidemiological study, means to predict the occurrence of particular disease. And finally, it is also helpful for disease forecast and management. So this is brief about detection and diagnosis of viruses. Next, the classification of detection and diagnosis methods. So this method actually broadly classified into two major groups. First one is conventional method and second one is the biotechnological method. So conventional method includes first the symptom-based diagnosis. So based on the symptom, based on specifically the characteristic symptom, we can predict that particular virus may be associated with that particular plant. Then biological method. So in biological method, host response, or it is actually use of indicator plant. Particular indicator plant will be used for detecting the particular virus. Next, cytopathological method. In cytopathological method, so virus actually induce some changes in cell or tissue. So at cyto, or means at cell level, the virus induced changes will be studied. For example, production of inclusion body. You might have heard about pinwheel structure, pinwheel inclusion bodies that are specifically produced by potato virus Y. So if a pinwheel type structure are present in a plant sample, then we can definitely say it was infected by virus. Next, the serological methods. So serological methods, it is based on antigen antibody interaction. Next, about the biotechnological methods. So these include the molecular methods. So molecular methods means the nucleic acid will be detected using PCR based method or loop mediated isothermal amplification method. Then nucleic acid hybridization method. So this method is based on hybridization between labeled probe and the virus nucleic acid. Okay. So by the hybridization, if it is truly hybridized, then we can confirm that particular virus is there. Then third method is advanced genomics techniques. So advanced genomic technique include the NGS, next generation sequencing methods. Next, the final one is biosensor and nanodiagnostic method. So using nanodiagnosis, nanotechnology or biosensors, we can detect the viruses. So we will discuss one by one each and everything. Okay. Next, first is the symptom-based diagnosis. So as you know that if a particular pathogen infect a particular plant, definitely it will produce some symptom. So by that symptom, by the production of characteristic symptom produced by virus, we can predict a particular virus association. Okay, like important symptom produced by viruses are mosaic, mottle, then chlorosis, vein clearing, ring spot, leaf curl, then stunting, necrosis. All these are symptoms produced by viruses, which vary from virus to virus. Tobacco mosaic virus, that will produce mosaic symptom. So like this we can predict. Then about the biological method, this is most important. Okay, so biological method includes use of indicator plant, host range studies, graft transmission and vector transmission. So here the virus are detected based on its reaction in host plant and transmission studies. So indicator plant are those plant which produce specific symptom, specific characteristic symptom when a particular virus is inoculated. Example of indicator plant is Chenopodium amaranticolor, this Nicotiana glutinosa, this is for the TMB. Okay, so indicator plant are mostly used for virus detection. Then host range studies. For virus, host specificity is very high. So particular virus can produce disease in particular range of plants only, on specific plants only. So host range study also helpful for predicting or detecting the virus. Then graft transmission. So graft transmission is also specific in nature. So this can be used for detection of plant viruses. Next, vector association. So vector transmission is also very specific in nature. Particular virus will be transmitted by particular species of insect only. Okay, so this can be also helpful for predicting the virus. Like aphid transmit the potato virus Y, CMV. Then this whitefly is major transmitter of this begomovirus. Leafhopper transmitted by grassy stand virus. So this can be useful. Next, about the cytopathological methods. So I have already told it studies the virus induced cellular changes. So that generally shows the that generally indicate the presence of the inclusion bodies. And inclusion bodies are nothing but these are actually virus particle only that are aggregated. These are actually excess particles that are formed during the replication pool. So they are removed from the replication pool and producing some inclusion body like structure which are maybe of different shape. So some particles are of like X shape, some are crystalline, some are pinwheel shape, some are crystalline is cylindrical shape. So based on its shape they are named accordingly, like X bodies, crystalline inclusion body. If it is producing the crystalline array, it will be called as crystalline inclusion. Then if it is producing star shaped structure or pinwheel structure, that will be called as pinwheel inclusion. If it is forming cylinder or rod shaped structure, then we will call it as cylindrical inclusion bodies. So these are different type of inclusion body produced by viruses, and these are produced by specific virus only. So by observing the inclusion bodies, we can predict the presence of particular virus. And this inclusion body actually generally observed with light microscopy, can be easily observed. Okay, and for further study means if you want to have a detailed study on plant viruses, then you can further go for electron microscopy, and specifically for TEM, transmission electron microscopy. Two types of microscopy is there: transmission electron microscopy and scanning electron microscopy. But this virus study can be only done with transmission electron microscopy. So various uses of this type method, like it can confirm the virus, useful for identification, for study and virus multiplication. These are normal things. Next, about the serological method. Serology means everybody knows that this is based on specific antibody and antigen reaction. Okay, it is based on antigen antibody reaction, the principle behind this, and the methods include. So these are the method, and you can classify actually this method into three category. Like first one is the conventional serological method. So conventional serological method includes this immunodiffusion and latex agglutination test. So these are actually conventional tests: ring precipitation method, immunodiffusion, then latex agglutination. So we will discuss all these. Are actually, this will take time. If I will describe these method, it will definitely take much time. So I am thinking to prepare separate video on serological, then molecular methods, and these things are already given in the laboratory techniques, so we will discuss there only. Then this is about the conventional and then modern serological method. So modern serological method includes the ELISA. ELISA can be of four types: dash ELISA means direct ELISA, indirect ELISA, dash ELISA or double antibody sandwich ELISA, and competitive ELISA. Four type of ELISA are there. Then another advanced serological method includes LFA, that is lateral flow assay. Okay, this is a chip based method types. Okay, so this is about the serological method. These are actually highly advantages because of its high specificity and high sensitivity in nature. Therefore, for virus detection, mostly serological methods are used. Next, about the molecular methods. So molecular methods include the detection of the virus specific molecule. Virus specific molecule means here they will detect the nucleic acid by using various amplification based technique. Amplification based technique is the PCR, obviously. So in addition to PCR, you can actually divide this molecular methods into three categories. First is the amplification method. You can divide into two categories. One is the PCR. So chain of reaction will be there for PCR. But another method is isothermal amplification. This loop mediated isothermal amplification. Here in this method, what happened? The total reaction will be occur at a constant temperature. But in PCR, if you know something about PCR, you know that various steps are there and all the steps are performed at different temperature. First, the denaturation that will occur at 94 degrees Celsius, then annealing at 50 to 54 degrees Celsius or 52 degrees Celsius, then extension step. So extension step will occur at 72 degrees Celsius. So there are chain of reaction are there in PCR which occur at particular temperature. That's why the PCR reaction is performed, or it needs a thermocycler, means cycling of temperature is needed to perform the PCR. But this limitation was actually omitted by the LAMP, loop mediated isothermal amplification. Here the nucleic acid can be amplified at a constant temperature, that is 60 to 65 degrees Celsius, using loop primers. So this is complicated things we will discuss in detail in separate video. Okay, so two types of amplification method we saw here. One is PCR and other is isothermal amplification. These are very commonly used as because of its very high sensitivity, very high specificity due to the use of primer and probes, very highly reliable result it gives, and early detection also. Okay, next about the nucleic acid based hybridization technique. Hybridization means complementary sequence will be hybridized. So this method includes dot blot technique. Dot blot means on a membrane, first the nucleic acid extracted from the virus will be spotted over the membrane, and then this our label probe, which already the sequence we know, that will be hybridized. So based on its hybridization, we can check whether the virus is present or not. Then another method is Southern blot method, which is particularly used for detection of DNA. So it is used for detection of DNA virus, and Northern blot for RNA virus. So this is based on hybridization method. Next, about advanced biotechnological methods. So advanced biotechnological methods includes use of genomics, bioinformatics, nano-based tools, and rapid sensitive comprehensive detection and identification of virus. So generally it is more advanced than ELISA and PCRs. So this include, the most commonly used method is the NGS, next generation sequencing. This is a very high throughput sequencing technique for virus detection. So whole genome will be sequenced, and using the data generator we can predict, means by blasting. If you know something about bioinformatics, you can get what is genome, that nucleic acid blast, protein blast. So by genome blast we can check which sequence is there and which virus may be associated. Then using DNA or RNA probes. So by using probes we can also identify. Then microarray technique, CRISPR-Cas based technique, biosensor and nano biosensors. So these are various biotechnological techniques, advanced biotechnological methods used for virus detection and diagnosis. So in laboratory technique only we will discuss all these things, means NGS, about this microarray, what is CRISPR-Cas, means it is a site at particular gene we can modify. CRISPR and Cas. So CRISPR is the site, means it will guide the RNA, guide our enzyme, restriction enzyme, to the target sequence. And Cas9 is a endonuclease. Cas9 or Cas12 or Cas13 is also there. So this Cas is this restriction endonuclease which will cut the particular sequence. So these are actually gene, means site specific. We can do the cleavage and silencing of gene. So these are various methods used for virus detection and diagnosis. So this is about today's video. So we have covered the detection and diagnosis methods used in plant viruses briefly. So in detail we will discuss, means what is ELISA, what is PCR, microarray, this NGS, everything we will discuss. This needs actually a separate video. I will definitely make separate video on all these things. Okay, so this is all about today's video. Thank you for watching.